And this will be the R side of the data viz story. And what's cool is that there's going to be some genomics data interwoven in this, which I think will be a really nice addition to the week, one of the most data-rich areas of ocean science that we have. So just before we start, I'll just mention that someone has just posted in the tutorials channel the path where you can find this in case anyone is still looking for it. And so that should all be available the way we did the tutorials yesterday, so you can follow along on Jupyter Hub. But feel free to, if you're having any trouble, continue posting questions in that tutorials channel, and we'll monitor it. Folks will try to answer, and if there's anything that I can ask to Maria as she goes along with her tutorial, I'll try to facilitate that as well. So without further ado, I'll pass it off to Maria.
Thank you, Nick, and let me share my screen. So yeah, I hope you're not surprised that there is a genomic section there, because it was not on the title of the of this talk. I hope to convince you that you should be interested in like genomics data for a variety of reasons. So they are, I mean, if you are working only, if you are only interested how like the sea moves, yes, then it might not be for you. But if you are interested in any forms or like biophysical interactions in the ocean productivity, biogeochemical cycles, distribution of nutrients, microbes especially play like a huge role. This actually microbes also affect the entire food web and the productivity and the fisheries. So all the omics of the ocean might be relevant to your research, and your research might be greatly benefit for implementing a genomic component. And we are, I think, living in a very exciting era that it's genomic data actually are available for oceanographers. Most of time series they have put the a genomic component in the data they are collecting. There have been in the past few years global expeditions. Tara is one of the most known ones. Malaspina as well. There have been developed remote access samplers, which are devices that they are placed in specific parts of the ocean. They collect samples either in time series or in specific events, and then they are processed in the lab to get omics data. And there is also development of genomic sensors. So I think we are going towards like bridging more the biology and omics with the typical oceanography, where you might have like a sensor in a mooring that doesn't only collect physicochemical data, but genomic data as well. And I think the possibilities are endless. But at the moment, it is very, very challenging to find them. Even like myself, that I am a microbial ecologist, and I work with those data. In most of the cases, they are deposited in raw formats. So people that they are data analysts cannot easily work with them. And as a field of like biological, like molecular biological oceanographers, our field is a little bit lagging. It took us like the first omics data started coming in the beginning of 2000, and we had to figure out many problems on how to quality control them. The sequencing platforms changed. So we had to develop the methodology to get from the sequences to the numbers and to the big data analysis. So a few disclaimers. There are many types of omics data. For this tutorial, we will work with the end result of the analysis of the sequences. So we will work with tables that they have numbers that represent species. And I will go into more detail when we are like viewing these tables. And if you're interested in the previous part, in the sequence analysis, there are many nice tutorials, and I would be happy to point people towards them. There are many different ways after that to process tables and to plot the data. You saw like amazing packages already. And because our field is lacking, we do things in a more like primitive way, in a more archaic way. So for SAP, and I didn't, by training, I'm not a data analyst. I try to become, because I see the opportunity there. But like my code is definitely not the prettier or the most efficient. And for those that they are advanced in either R or Python, this tutorial might be like basic for them. So I think that the previous one we had the advanced postgraduate like lecture. Now we have like visualizing R 101. But I want to for you to start thinking, because the data of like the molecular genomics data are already they're kind of multiplexed. So we don't get like we don't get one value. We don't get like temperature. We get values of several species that they are in the ocean. So they are more complex. They are complex structures. Most of you, like the physical oceanographers, know how to deal with data. So for me, this tutorial is most of like opportunity for you to understand what kind of data we are producing and how to put together, like to bridge the worlds and make most out of this data. So the amplicon data is the most common type of environmental omics data. And those actually came from small fragments of the genomic material. So we read those fragments and we are able to make, to tell who is there, the identity of different microbes, and we give some numbers with that. I will talk about the numbers a little bit later, but we cannot make absolute estimates. So we cannot make estimates of how many cells are per ml of seawater, because the more we sequence, the more like the more sequencing effort we put, the more number of sequences we will get. So we tend to work more with relative abundances than the absolute numbers. And we have to keep that in mind as we go. And I have some additional resources about this amplicon processing, and then a link to a bioinformatics virtual coordinator network that was kind of self-organized, and Ben Pali led this effort that created an online learning community during COVID for graduate students and students who work with omics data to be able to learn and work with their for their projects. And in some of the other the other tabs, I have some basic R commands, but I cover, let me see, yes. But I will cover them during the tutorial as well. But if you forget and you want to see in which working directory you are, to list files, to create a directory, I have some simple commands for you. And we can now go to the tutorial on how we like traditionally plot omics data, and I hope that will give us some food for thought. So we can start by loading our packages. Let's see, we're here. Maybe my kernel is, let me refresh that. Yes. Any of the other ones, I can see that they are, oh, I think. The clean version, I think, stopped working. Maybe. Yeah. Okay, so if you have any questions while I'm waiting for my terminal to load, let me know, Nick.
Yep. Ben is saying Maria needs to assign a kernel to the notebook. And we're also hearing.
Oh, you're right.
We're also hearing people being happy to get visualizing R101. We've got the right audience.
Okay, let me.
I think you need to go to kernel at the top menu, like you have file edit. Yes, that works.
Yes.
Perfect.
I was pushing the wrong. Okay, sorry for that. Okay, we load the packages, and there are actually. Sorry. There are actually packages that can plot the data for you. While we were, Felipe was putting the R kernel together, we chose to go with the basic, not the basic, with packages that they are already in CRAN. So other type of packages like Filoseek or Amphis2, that they will do the processing for you. You can get them from GitHub, and it might have been a little bit more challenging to implement, but we will produce all the graphs we need with those basic packages. Ignore the warnings, nothing serious. So I like to use some of the palettes that the color brewer that we loaded up here has, and we can see pre-existing palettes here. There is a limited amount of colors that exist, so you have to be careful when you color and you want to color more groups than the actual colors that they are offered, but they come handy sometimes. And I can set now the palette that I will be using for the plots that will come a little bit later. So the data set we are going to use, it's an Amplicon data set of the bacterial communities from Cariaco Basin, which is off the coast of Venezuela, from a recently published paper with some colleagues of mine. And this is a stratified water column. And this stratification actually leads to anoxia in the bottom. So we have different layers of water that when we did the collection, we actually were monitor oxygen. So we sampled from oxic to disoxic, suboxic, anoxic, sulfidic, and euxenic. So there is a gradient of oxygen, and we believe that we know from microbial physiology that those would should alter the communities that they are there. And we sampled to different time points, May and November, because we wanted to see difference between the productive upwelling and non-productive season. So let's go here and import our output table. And the output table, as you see here, will be inside your input data. But we don't have to go there to visualize it. We can verify that it has become actually an object in, yes. There is an object in R actually, and we can go with the head command to see the table. And this is the typical format of the tables that genomics data produce. So we have an ODU ID here, which is an ID for the species, for the different type of microorganisms that they are there, and the names do not mean anything. It's how they come from the software that processes our data. And here we have the each column is a header of our samples, and then at the end there is usually taxonomy. And taxonomy actually it is the one that gives us the identity of the organism. And as you see, the taxonomy has different ranks. So bacteria is the big group or the kingdom. Proteobacteria is the phylum. Gamma proteobacteria is the class. And we need these taxonomic rankings in order to be able to parse the information in different levels. You will see it a little bit later. And I want to briefly show you the tail end of the table, because as you will observe here, we have a lot of zeros. And that is a problem of the method actually, or the fact that we can go very deep with sequencing and recover some species that they might be rare. But we have to be mindful when we are doing this analysis. And I have, in the following lines, I have quickly summarized the number of columns that they have 0 entries for each one of the samples. And then here I have actually calculated the proportion. So if you see sometimes 93, like 93 of our rows, they contain zeros. So like most of the species are only found in specific in specific samples, and it's absent in the other ones. And a way to quickly visualize our sequencing effort and the richness of species is to plot the rare curves. And the rare curves actually does exactly that. Plots the number of species as a function of the numbers of reads we sequence. And I will fire that because this actually takes a little bit time in order to calculate. But as you will see it, we will get an idea whether our sequencing effort was good enough. And ideally, we want to reach, oh, they're here. So those are not actually great. We want to reach like sequencing effort. As you see, the number of reads is here. We want to reach the effort that our curves is entering a plateau. And for these two samples especially, our sequencing effort was not enough in order to be able to recover the full diversity. And if you see a little bit, let me try to maximize my screen a little bit. So if you see the two greens, oh yeah, there is a little bit of lag. The two green ones, they are actually they are actually the samples of pharoxic samples, and both of them show the same trend. So those have the richest genomic diversity there. And it's always good to have this as a first pass of our analysis to see how far we can, how far we have sampled if we want to sample more. And then in order for the plotting, we'll mostly use ggplot2, and I think that in following tutorials, you will be using this package in order to plot. I found it very, very easy. It creates beautiful, beautiful plots. The format that I find that works better for ggplot2 is the long format, and we can talk. At some point after this tutorial about the long versus wide format in the data. but I will transform my data here in the log format, and at the same time, I will do a couple of things. I will parse the sample headers to associate its sample with what we call feature. So, and the feature refers to what kind of, like how to the oxygen there, the month, and the depth. And they are going to separate the taxonomy to the different taxonomic ranks. So now, if you see the head of my table, I have the OTU here, the taxonomy, which is broken down to different taxonomic ranks. Then the sample name, some like kind of metadata for the sample, when I parse the header here, and the actual count. So in the previous format in the wide, I had the counts as a matrix. Now its a row is actually one count. Do we have any questions, Nick, so far?
I think we're good. There were a couple of questions about where to find the tutorial, but I think everything has been answered on the Slack channel so far.
Perfect. Okay, so I will just briefly show people how the dimension from the white table to log chains. And then in order to start plotting, because I cannot, it's very, there is no way that I could visualize the plot with individual species, I will be grouping the my data, and I will group them in different levels. For starters, I will group them in the kingdom level because I want to see if I, although I have been aiming only for bacteria with my methods, whether I got sequences from other kingdoms, which happens very often, or I got, as in this case, what we call no blast hit. So we were not able to identify with the existing database what kind of bacteria was there. And if you see for this data set, the number actually of bacteria is rather low. So it's not necessary to remove them from the subsequent analysis, but I will be excluding them from our plots. And then because in tables, it's better to work actually with NAs rather no blast hits, I will briefly do so. So I will substitute here the no blast hit with NAs. And now I will summarize the counts to the phylum level for its sample. And I will filter out the ones that they are NAs in the kingdom level. Again, you can ignore the warning. If we have, I think, the tidyverse has it's a different version from the one that I was using was when I was putting in the commands. So, but they work properly. So now for each one of the samples, you can see the counts of each one of the phyla. And we are ready to create our first bar plot to see the data. And this is one of the typical bar plots we get, but as you might already notice, we have so many colors here that they're very hard to distinguish this from that, but we don't see many colors in the plot itself. So as I mentioned, like bacterial diversity is huge. So yes, we have a variety of different phyla there, but the ones that make the majority of our data, it's only a handful, most probably. And here we have plotted one sample after another, and it's very difficult to see, like, and to be able to order them in a nice way. So here using like the facet grid, which is a possibility that ggplot offers, we can actually separate the bar plot from our May sampling to the November sampling. So that started looking a little bit better, but still we have a lot of phyla here. So with the next 2 commands, I actually filter my data to get the phyla that the 10 most abundant phyla that we see here. And I will be plotting only then the 10 most abundant phyla, as you see here. If you compare the previous plots with this one, you will notice that we don't seem to be missing anything, but I have a small exercise for you that you can work on your own and let me know if you have questions in order to be able to calculate how many, like the percentage of reads that we kind of lost is not represented in this bar graph. And for now, I was not very happy with the plot as is, for a number of reasons. My axis titles are not great, and also the sequence of the samples is not great. As I mentioned, we were sampling from the surface, the oxygenated sample to the euxinic, and here ggplot plots the categories in alphabetical order, so I don't like them to be messed up, and I will change that, and before changing that, I will change that, and at the same time, by changing this stats, so here in the previous one, we have the position stack that created the absolute numbers, and here we go to the position field, and we have the relative abundance of the phyla, actually. And that looks much better for me. The samples are in the order that they were actually sampled, so they make much more sense. And another small exercise for you is to see if you can reverse the axis. So in oceanography, sometimes we like to visualize things from the oxic layer or from the shallow layer to the deep one. And this is a fun exercise that you can try at home. And of course, there are ways that you can easily change the colors you need using the palette we have loaded before. And the small like remark that I want to make here on how to understand the microbial data, if you see the outcome of this plot, we can definitely see that the trend is changing. So the relative abundances of the phyla definitely change for the deepest euxinic sample. But for the other ones, especially the pink color, which is proteobacteria, seems more or less to be constant, like for all the depths, the oxygen features, which like biologically doesn't make much sense, because you go from samples that they were taken in 100 meters to samples that they are taking 250 meters with drastically change of physicochemical conditions. So this rank might be too coarse, too rough, too general in order to give us the difference in patterns. And for this reason, I am going to explore more deeply what happens with the proteobacteria only. So in the previous command, I have been selecting for Proteobacteria only. And I am plotting here the different classes of alpha Proteobacteria. And as you see, we start seeing differences in patterns of classes of different cyanobacteria. In the first 3 depths, we see almost a codominance of the alpha Proteobacteria and gamma Proteobacteria. And then here we have a dominance of gamma Proteobacteria. And if you go further in exploring the data, you'll probably realize that there are also groups within gamma Proteobacteria chains. And another, do we have questions in the first part, Nick?
Nope, just some commentary going along, people sharing resources and that sort of thing.
Great.
So, yep.
Yes. So as I mentioned, this is a rather small data set. So it's the data set itself. So the amount of sequences that I'm presenting you, like the table, is the full table that that we used for the publication. I just excluded some samples in order to make it easier for us to visualize. But if you want to sync patterns of a global expedition of a time series, these bar plots are not as informative. After, if you have 100 or 200 samples, it would be difficult to observe like differences in patterns. So in microbial ecology, we use ordinations to reveal patterns of distributions and to explore whether environmental factors might be shaping these microbial communities or be shaped by them. Like the multivariate analysis and ordination is a big chapter on itself, and it's more challenging in microbial ecology for a variety of reasons. One is that our data is not usually normal. So if some tools, they assume normality of data that doesn't exist most of the times. There are so many different methods to make ordinations. And we also have in the tables, the rows that have many zeros. And because these ordinations are based on matrices of dissimilarity or similarity, we need to take into account that when you have when samples share zeros, that doesn't make them similar. It can be, it is a bias of the method, and also it doesn't reflect biological similarity. So our data usually need transformation, and again, I have provided people links because this is another huge chapter, but one of the most commonly used is a Hellinger transformation. And we can briefly apply that and then go and do the principal component analysis. For the principal component analysis here, I'm presenting the major components. So there are many axes, and the principal component analysis, as all multivariate analysis, try to explain the dissimilarity by placing our communities into a multidimensional matrix. And here I briefly visualize how much of the fraction of the variance that is explained from each one of the axes. No, I have to, I'm not sure actually. I'll get back to you about that. what I will skip this part because I just visualize the axis. But here, when we visualize the PCA, I first make a data frame of the values that we have seen before. And then we can plot this. Oh, and he doesn't like, yeah, let me. Something that I used here. Okay, here is my ordination. So actually at the previous step, I transformed the numbers that I got from the axis to the percentage that shows how much of the variance between the communities is explained by each one of the axes. And actually these percentages, they are very high. We don't usually see them in the microbial community. The two axes together, usually we can explain up to 20%. These particular samples, because it's a stratified community and the community changes very dramatically, give us these nice separations. And these samples here are not ordered by the oxygen regime, but we go here from deoxic samples to disoxic, to suboxic. So like mentally or biologically, the representation that we have makes sense. And keep in mind that each one of the points, it is the similarity of the entire communities. So for the presence and absence of all species that they are there. And I will briefly try to fit the environmental factors that I have in this table here. And some of the metadata is oxygen, depth, temperature, salinity, hydrogen sulfide. And I see here that I have only two factors that they are statistically significant for explaining the variance that we see above. And I use the output of the analysis to convert it to data frame in order to be able to plot it. So now I have again, the previous ordination. I have just plotted the environmental factors that they are driving actually the separation of the communities. And the euxine example is the most, the deepest one, the one that has the highest hydrogen sulfide, and that clearly differentiates this from everything else. So for the interest of time, as I told you, we work with a small data set there, but I looked for large data sets, and Tara Oceans, for most of their work, they have only published the raw data, the sequences, but for the microbial eukaryotes, they have released their databases. So this is the table that has the OTUs, which is the different species that they have sampled, and this is the parameters. So it has the location and the environmental parameters. So I would challenge you to download this data, and if you are interested in marine omics, try to play with this data, try to create the same plots and explore them further as you go to more advanced tutorial, and start treating like the groups of organisms or specific species as one of the physicochemical parameters that would be that you would be plotting with other software. And I know that Nick and Ben and Camille might have a tutorial that takes into a biological component. And I can stop here for questions, and I have a couple of more things, but we are running out of time. So here is the creation of heat maps with similar type of data. And I can also show this one, which is the primitive way of biologists to create, oh yeah, I didn't know. So it's a primitive way of biologists to create actual maps of abundances. It's not very, it won't take a long time, but you will be creating you will be creating maps in a more elegant way. So I would challenge you to try to create a map like the more advanced maps. So here is, and it's I couldn't make it too precise. So here I have plotted the relative abundance of a specific genome in the data in the terameta-genomes, which reflects its relative abundance to the environment. And you can see its distribution. So yeah, physical oceanographers and Felipe might be laughing with this map. I laugh as well, but it was published. So I'm one of my, of my goals of this week is try to catch up with all the cool tools and cool packages that you are using. So hopefully we can bridge these two well. And yes. So let me know if you have questions or if you want me to discuss any of the tutorial aspects more.
There's no questions on the Slack channel, Maria, and you do have a few more minutes if you wanted to show the heat map example or yes, anything else that you didn't yes, get.
I can definitely show the heat map example. So and I find heat maps like when I first saw this heat map and the this data set here, it's from another publication that Nick was a co-author actually, and we tried to, we were interested in a specific functional group of nitrite oxidizing bacteria, and from other experiments we had seen that they were very important in their role on the carbon fixation in the ocean. And carbon fixation is a big topic in the Mesopelagic because it actually it influences the carbon pump. CO2 is fixed, is removed from the water, and they are also major players in nitrate in the nitrogen cycle. So they do both amazing jobs for the ocean. And we used genomes that my former postdoc supervisor, Ramona Stepanauskas, has sampled. And in order to see how to estimate their abundance in the ocean, we took all the tarameta genomes from different depths and different locations. And we used the metagenomic reads against our genomes to see how much of the metagenome actually we could explain, how much of the metagenome was reflected in those SARs. And this is a long table for more of the columns we don't need to worry about. We can plot any of the proportion of the total reads of the total base pairs that they are recruited. And at this point, I think at the beginning, I was using one of Ben's software that it was designed for, it was designed for another application, actually for another sequencing technology. So the Julia, who is a bioinformatician at Beagle as well, if we reload my instance, you will see, or you can just fire the commands. So making heat plot maps creates usually awful results. So if you use the standard ggplot2, you will get this black, almost black, that says that nothing is here. So we did a couple of tweaks, and by using a logarithmic scale, warm colors for the high abundance, like cold colors for the low abundance, we start actually seeing patterns. And this heat map is not in the in its final format, and I wanted to show you why, because we started seeing these patterns of high abundance, the rest colors, and if you go and see the legend here, these are all mesopelagic samples. And then by clustering SAGs and metagenomes here, you end up with a nicer map. Here I challenge you to write your own code. Try to cluster the SAGs here, or the genomes here, or the metagenomes here, in order to create like groups of similarity. But if you cannot achieve that, Julia has wrote a beautiful script that is, and I have given you the link here, that it will do this for you. And yeah, and I took one of these genomes from here and plot its abundance actually to the map. And of course here you lose the depth perspective. And this is why I think it is, you can use different colors if you want to show that. But I would like to find a more elegant way to create maps.
Hey Maria, there is a question now. Hang on, let me pull up. Is there an easy way to make a triangular heat map in R, like for correlation matrix?
And I don't know the answer to that. Maybe Nick or Ben, that has worked a lot with maps. But if we don't know the answer, I can definitely note the question and get back to you.
It's in the tutorial Slack channel, so you should be able to find it, and we can type replies in there.
Mm-hmm. Yeah, that would be, yeah, thank you for the question. I'll definitely look this up.
Thanks, Maria. Yeah, I would say there's probably something in ggplot that would make that pretty easy. I think the question is just sort of about a gridded, kind of like the heat map of a correlation matrix. So there are probably a lot of things you could do with that in ggplot. But yeah, but we can follow up in the tutorial Slack channel on that. So yeah, thanks, Maria. That was great.